thanks to those of you who've come along. This is the second in the Fluorescence Microscopy series, Introduction to Fluorescence Microscopy. If you've been attending over the last couple of weeks, you may have come to the Intro to Fluorescence lecture that I gave last Monday. And we're down here now. So today we're going to cover applications in fluorescence microscopy, and then on Thursday, same place, same time, we'll cover experimental design, acquisition, and analysis. This is an overview of what we're going to cover today. I might just make the screen a bit darker so you guys can see a bit better. So first of all, we're going to cover BOMP. So I'm the manager of the Biological Optical Microscopy platform here at the university. So we're going to cover the who, what, and where of what BOMP is very briefly. We'll then spend quite a bit of time going through fluorescence microscopes. And what I'm going to do is explain the anatomy of a standard wide field or epifluorescence microscope and compare that against a confocal microscope, so you get an idea of how those two different systems work and what are the key differences between them. And then the last section of the lecture will cover the experimental applications. So basically, what's the cool stuff you can do with our equipment you might be able to use in your own research. So let's start with BOMP. So the first question is, what is BOMP? So about 7 years ago, the university realized it had a lot of microscopes, but they were all hidden in different buildings and in different labs, and somebody might not know how to use it or how to get access to it or whether it actually existed. So they established our platform to basically be a central point of coordination for all those pieces of equipment. So we're now up to 21 high-end fluorescence microscopes, and a few analysis computers in there as well. And we're dotted around in different locations with different types of equipment. This is an overview. I'm not expecting you to go through the content of that. So one of the first things we did when we established the platform was to set up a centralized booking system. So you can sign up to the BOMP platform. This runs on the Bi-21 booking system. And all of those microscopes and analysis computers can be found in one place. So you can search, see which ones are available, which ones might be within your building or close to you, and then book those online. The other thing we've established is a centralized website as well. So we have this microscopy.unimelb.edu.au webmail, internet address, web page address. It's shared between us, which is optical microscopy, and the electron microscopy facility as well down at Bi-21. And I spend a lot of time keeping this up to date, so any of the events that we have go into the news and events section so you guys can link in. I might just quickly come out and go to the web page and just give you a live demo. So as I said, we're under the optical microscopy tab, and we've got links here for new users. So if you want to get trained on one of our instruments, you can go into the new user section. That's broken down. The web page works by location. And there's an online training and registration page for each location. We have a similar table to the one you've just seen with all the instruments, again broken down by location. Here we have specification sheets for each of the instruments, so you can see what objectives, what lasers, what detectors and things are on each of the microscopes to see whether they fit your experiments. There are links to the calendar so you can book them. There are our user agreement forms which have the charges essentially for how much it is to use those. And then we've also got the links again also if you want to register for training on those instruments. Let's go back in. So the last part of today's talk will be applications. We also have a web page based on this as well. So it's not an exhaustive list, but there are a lot of different applications that are possible. You can click on one of these, So it's like a high content screen. It goes in, gives you a brief explanation, and then tells you what system is capable of doing those types of experiments. There's a link again to the booking calendar, which you've already seen. We've got a frequently asked questions page. So some use to you guys is probably the online resources section. So all of the content from these lecture series and all of the events that we run go into this workshops and resources tab. So probably next week I will load up the intro to lecture series into this region here. There'll be a new line here with all of the links to the PDFs of the talks I'm giving, and also the lecture capture as well. So these are all being recorded. If you want to do some image analysis, we also have Fiji workshop resources. So we've got our Fiji for Beginners workshop running this afternoon from 1 till 5. If you can't come along to that, then all of the resources for that are online, so you can download again the presentations, the demo data we're using, so you can go through it at your own pace. We also have a YouTube playlist, so I've gone through and recorded each of the things we do in that workshop. And if you can't remember how to add a scale bar, then you just come in, find the calibrating an image, adding a scale bar, and there's a 2-minute YouTube video which takes you through how to do that. So we've got that for the beginners workshop and also the quantification workshop. We have YouTube videos there. We also have a Fiji macro routing workshop. We haven't yet recorded this, but you can download the workshop content there as well. Okay, so that's just a quick, brief overview of the webpage, what you can find there. Let's get back to the presentation. So I mentioned we were distributed across the university. We are currently in 6 different buildings across the university. So here in the medical building, our main facility is through that wall. If at the end you guys want to have a look through the lab, I'm happy to take you on a tour through the facilities we have here in the medical building. We're also located at the Bio21 Institute on Flemington Road, Peter Doherty Institute, which is just across the building sites. Biosciences 2 and 4, which are further up on main campus. And then we're in a couple of the hospital locations, Royal Melbourne Hospital and also St Vincent's Hospital as well. The main thing that's important to you guys, I guess, is probably who we are and what we can do for you guys. So my name's Paul McMillan, I'm the manager of the platform. I also have part responsibility of looking after the Bio21 Institute and the Department of Medicine here at Royal Melbourne Hospital. And my expertise is in super resolution microscopy. Ellie Cho, who sits in the office just across the corridor here, she looks after our high content imaging system and is also our image analysis guru. So if you've got any curly image analysis questions, she's the person to go to. Hamid looks after Eastern Hill Precinct, Biosciences, and a new node which is about to come on, which is the School of Physics. And his background, he's got a PhD in fluorescence lifetime imaging and also is skilled in fluorescence fluctuation spectroscopy. A lot of these high-end techniques I'll discuss in the applications section. Sam Mills looks after our medical building node, as I said, through the wall there, and his expertise in confocal and light sheet imaging. And Eleanor is primarily looking after the Doherty Institute, but also a little bit medical building, and her expertise is in confocal microscopy. If you don't know who to approach or how to approach them, then we have a general BOMP inquiries email that you can send any of your questions to, and we'll get the right person to answer your questions. So that's the team. We're employed by the university to help the university's researchers. So if you have any questions, obviously more around microscopy, then please feel free to ask us, and we can try and help you out as much as we can. Ways that we can help are things like consultation. So we can help from the very start to the very end of your experiment. So experimental design, how you're acquiring the data, and then finally how you're interpreting the data at the end. We do a lot in training people. So we've got seminars and workshops that cover the theory and applications in fluorescence microscopy. We have over 700 people a year attending those training events. We also do over 250 instrument inductions a year as well across those multiple instruments. If you're fairly new to fluorescence microscopy and your boss has asked you to do, a fairly involved technique, then we do collaborations where, as we're the experts in the field, we can help to kind of push and develop your project as it's going forward. So if you're trying to replicate something you read in a paper but you don't know how to do it, then we can work with you and try and get your project up and running. So this is on more the high-end techniques, so things like super resolution microscopy, you work with me, Flume and FFS, you work with Hamid, light sheet, you're working with Sam, and high content imaging, you're working with Ellie in that case. And normally we ask, because it's collaboration, it's just essentially a scientific collaboration, if we're improving the project or any publications that are coming out, we expect to then be authors on those publications. Image analysis. So as I mentioned, we do a lot of training workshops. Ellie also will write specific code for people to do automated image analysis, and again she does that on a collaborative basis. And we can also help with things like manuscript preparation, so how to present your data, how to analyze your data, how to interpret your data. And also we can help with grant writing as well. So either for research grant applications, we can read it, make sure it makes sense, suggests different terminology or how you might approach a problem. Or we're also quite well versed in writing equipment grant applications as well. So if you're interested in those, we can help with that as well. Okay, so that's our basic introduction to the platform. I didn't want to spend too much time on that. We'll spend most of the time going through fluorescence microscopes. And as I mentioned, we're going to compare a wide field or epifluorescence microscope against a confocal microscope. So on the left image here, we've got epifluorescence image, and on the right, we've got a confocal image. So our platform tends to be confocal and above. We don't have very many basic epifluorescence or wide field systems. They tend to be a departmental or a lab resource. We can still advise and help you out on them if you're having problems with your own systems. Okay, so let's go through your basic epifluorescence microscope. This is an extremely complicated cut through of what an epifluorescence microscope looks like. We're going to go with a slightly simpler version. I've highlighted 5 key areas within the layout of that microscope. So the first thing is the illumination, so the light source that we're illuminating our sample with. Next thing we have is an excitation filter. The next and most important part of your microscope is the objective. After that, we've got, we'll call it dichroic mirror, but also an emission filter as well. And then finally, when you're taking and acquiring an image, we've got some kind of detector on an epifluorescence microscope. That tends to be a digital camera. And we'll go through each one of these 5 steps and explain what are the key different components. So firstly, illumination. What are we illuminating our sample with? So if you remember, we're imaging with fluorescence. We're trying to excite fluorophores at specific wavelengths that are going to match the excitation spectrum. So there are multiple ways we can do that. Some of the older methods use bulbs that contain things like mercury or xenon. More recently, these have switched into metal halide bulbs. And even more recently, people are starting to use light-emitting diodes. So I'll show you a few examples of these. These are mercury arc bulbs, so you may have seen some of these on some of your own systems. They produce essentially white light, so they produce wavelengths across the entire visible light spectrum. There are a few peaks within that, but essentially it's going to generate white light coming out of the system. One of the downsides of these mercury arc bulbs is they don't last very long, so they're rated for about 200 hours. After that, you've got the risk of the bulbs actually starting to explode, and they tend to get quite dim towards the end of their lifespans as well. One of the reasons that people have switched to LEDs is that you can tune them so that they're basically a very narrow band of light that's coming out. So in this case, we're emitting a very narrow band at about 560 nanometers in that green light range. The other positive things with LEDs is they have a much longer lifespan, so they're into the kind of tens of thousands of hours rather than 200 hours. So you'll find a lot of the more common, more recently released systems are using LEDs for illumination over the traditional bulb. Next thing is our excitation filter. So if we've got white light coming into the system and we only want to excite a single fluorophore at any one time, we've got to block some of that light. Okay? We've got to select the wavelength that we're going to excite our sample with. And we're doing that with what we call an excitation filter. So here, if you can see, we've got white light coming in from the right-hand side. We've got a filter here, and it's filtering everything out apart from the green light. And this is what we call a bandpass filter. So here, it's only allowing the light in this band here, in this green range, to transmit through the filter, blocking everything else. So you're selecting just the wavelengths that you want to excite your sample with. Now, in most microscopes, these will sit, so this is your excitation filter here. These will sit in what we call a filter cube. And this filter cube is made up of 3 different parts. So we've got our excitation filter here. We've got a dichroic mirror, or some kind of, that's called a beam splitting mirror here. And then we have our emission filter at the top here. So what's happening in this case is we've got white light coming in, being filtered out, so that just a specific wavelength is traveling through that first filter. This dichroic mirror then reflects that light down. It's going to go down to your objective, through the objective, and onto your sample to excite the fluorescence. Once your fluorophore has been excited, if you remember, it's emitting in a longer wavelength than it's excited because of the Stokes shift in fluorescence. So you lose a bit of energy, so the wavelength increases. And so that increased wavelength is emitted out from your sample, collected by the objective, comes back up, and because this dichroic mirror is especially set up, it's set up to reflect the excitation light, but allow the emission light to go through. So it's a specially tuned mirror which reacts differently to different wavelengths of light. That allows that light to transmit through. We can further refine what light's getting to our eyepiece or our detector using this emission filter. So we've got 3 different components, all sitting in this filter cube, which is sitting between the illumination and the sample and the eyepiece. Okay, next thing is our objective. So as I mentioned, this is the most important part of any microscope. Please, please, please take good care of your objectives. We've just had one smashed in the School of Biosciences, and it cost us $8,000 to replace it. And that's because someone didn't realize where the stage was in relation to the objective and drove the stage right into the objective. Smashed the front glass part of this, and we had to buy a whole new objective. Okay, everyone looks at the objectives and sees all this writing, doesn't really know what's going on or what it all means. But there's actually a lot of very useful information written onto the objectives. So let's start at the top and work our way down. I'll just go here. So first thing at the top here, we've got any types of corrections that we might have on the objective. So here we've got flat field correction, so how evenly illuminated is the field of view. If this is uncorrected, obviously it's not going to be as nice as if it has been corrected. In APO, we've got chromatic correction. So what you'll find in an uncorrected lens is that your green channel might focus at a different point to your red channel and might focus at another different point to your blue channel. On a well-corrected So like an apo-corrected lens, you'll find that they'll all focus into the same point. So you want to check what type of corrections there are on your objectives. Next thing is magnification. Again, we've got that number here, but there's a couple of important informations after that as well. So this number that comes after the magnification is 1.40, and this image is what we call our numerical aperture. And that's what's giving us the readout in terms of the resolution of your objective. So resolution is actually irrespective of the magnification. It's tied up by the numerical aperture. And the next thing we've got is what type of immersion medium is it? So is it an oil lens, in which case it's expecting oil between the front of the objective lens and the cover slip? Is it an air lens, in which case it won't say anything there? Sometimes you'll see water, sometimes you'll see glycerol. Some lenses have correction colors which allow you to modify what immersion liquids you're using. Next thing is cover slip thickness. So this number here is 0.17 is telling you what thickness of cover glass that objective is expecting to be in the light path. So it's optically set up to match with a preferred cover slip thickness. Normally this would be a number 1.5 cover slip, so this 170 microns is 0.17 of a millimeter. Again, some objectives have correction colors that can actually change this, but most systems will be expecting that number 1.5 cover slip. If your lab has a stack of number 1 cover slips, when you get back to the lab after this, please throw them gently into a glass bin and order in number 1.5 cover slips. That is what most fluorescence microscopes will be expecting, and you'll get better image quality, especially in Z, as you're going deeper into samples, if you have that 170 micron thickness cover glass. Another really nice thing that a lot of people don't notice is this color around the objective is actually color-coded by magnification. So on some inverted microscopes, it's really difficult to get your head under the stage and read what objective is in place. But normally you can see this color here, and depending on the color of that, that will tell you what the magnification of the lens is. So 5x is red, 10x is green, 10x is yellow, 20x is green, 40x is light blue, 60x is dark blue, and 100x is white. Okay, so I mentioned about taking care of your objectives. Some key points here. So air lenses obviously are not designed to come into contact with oil. It does happen on a regular basis, we understand that. But if you know that you accidentally have oil on an air lens, then please contact whoever's responsible for that as soon as possible. If that's one of our instruments, please call us and we'll come across and clean it off. Not that it's a massive emergency to take the oil off the air lens, but it needs to be done properly. Sometimes it can be quite tricky to get the oil out. And also, if you don't let us know immediately, then it can affect the next people who come to use the instrument. So we get regular telephone calls saying, I can't see anything on the 10x objective, everything's blurry, I can't focus. And 99% of the time it's because the previous person accidentally got oil onto the lens. So if you know you've done it, we're not going to shout at you, just let us know so we can come and clean the lens off. For oil immersion lenses, in terms of cleaning them, please use specialized lens tissue. So within our platform, we provide that to the users. Don't use Kim wipes or tissues, as these are too abrasive for the lenses. I normally do a 3-step cleaning procedure. So I'll take a strip of lens tissue and I'll basically gently dab it on top of the central glass part of the lens, just trying to mop up as much of the oil as possible. If I do any motion, then it's normally around the metal outside part. I won't press down and move across the central glass part because you can get bits of dust trapped and you're basically etching into the glass on the front of the objective. The second part is to soak a piece of lens tissue in some kind of solvent. So we normally use 70 or 80% ethanol for this. And normally if this is my objective and this is my lens tissue, I'll pre-soak it, I'll sit it down, and then I'll gently just pull it across. So there's no downward motion, we're just gently allowing gravity to hold it on, and we're just pulling that across. And then the third step, again with a dry piece of lens tissue, is just to go back and clean around the outside and take off any excess ethanol that might be around. So just be very, very careful how you're dealing with those objectives. Okay, we've already covered emission filters a bit, so these tend to sit in the filter block immediately before you're getting to the eyepiece or the camera. And this is again you selecting what wavelengths we want to visualize either through our eyepieces or take a picture of in the camera. And again, it's normally specific to a certain fluorophore, so like a GFP filter or DS red filter or something like that. But you can get different types of emission filters. So we have short pass, long pass, and band pass. Now we've already covered band pass on the excitation filter. It's a narrow band that you're selecting. I'll show you an example of a long pass filter. So this is, sometimes you might see in our specification sheet, something like an LP520. LP stands for long pass. It basically means it's going to let anything above 520 nanometers go through that emission filter. Okay, so you'll see all of these wavelengths coming through. I mentioned band pass already. We can get dual band pass filters. You can have a filter that allows two bands of light to go through. This is very often for like a live cell setup where you don't want to switch filters, emission filters between acquisitions. Normally these would be on diode illumination systems where you can switch the diodes on and off quite quickly, so you're only exciting either the green channel or the red channel. Okay, then the final step in the epifluorescence system is the camera. So this could be an hour's lecture in itself, all the different types of cameras and possibilities. If you are buying a new microscope, then there are a lot of considerations that you might make, and a lot of it comes down to the applications that you're going to be using the systems for at the end. There are different types of digital cameras available, so CCDs, charge-coupled devices, electron multiplying charge-coupled devices, and scientific CMOS. That's the cameras that are sitting in our phones at the moment. Each of these have different pros and cons. So scientific CMOS tend to be quite fast. EMCCDs very useful in low light environments. Some of the things you have to take into consideration when you're purchasing a new camera is things like quantum efficiency. So how efficient is it at converting photons coming into the camera into an electrical signal? Things like pixel dimensions, am I going to be able to sample correctly? And we'll cover sampling later on in the talk. Monochrome versus color. So if you're imaging only in fluorescence, then you would normally go with a monochrome camera, and that's because you're using filters basically to block out the light. You're just counting photons basically coming through based on the emission filters. If you're doing something like a H&E stain or something like that, then you're probably going to need a color camera. You can acquire fluorescence images on a color camera, but you decrease your resolution and your sensitivity quite a bit by doing that. There are companies now which will sell dual monochrome and color cameras, so you can switch between the modes if you want to. Final consideration is bit depth. This is basically the number of grayscale values that you separate your image out into. So if you're just imaging to generate an image, then something like an 8-bit camera is probably fine. If you're trying to quantify the data, then you probably want to stretch and get like a 12 or a 16-bit camera. So you've got a little bit more information there, which will allow you to quantify a bit better. Okay, so that's the 5 kind of key parts of an epifluorescence microscope. Obviously there are thousands of components, but we don't have time to go through all of those today. I guess the second part of this is we're going to look at confocals, but I wanted to just explain one of the key differences between the epifluorescence and confocal microscopy. And it's shown in this figure here. So with epifluorescence, we're illuminating the full field of view, and not just in our current focal plane, but above the focal plane and below the focal plane. This means that we can actually see quite a lot of the out of focus light. So if you remember back to that first image where I had the epifluorescence image versus the confocal image, the epifluorescence image was quite fuzzy, whereas the confocal image was very, very sharp. And that's because on the wide field mode, you're illuminating everything above and below the focal plane. So it can get a little bit fuzzy on thicker samples. On epifluorescence systems, your best imaging is only going to be in the first 20 microns past the cover slip. Once you start going further than that, you're going to hit a whole heap of aberrations and problems that's going to make your imaging quite tricky. In general, you should always be preparing your samples on the cover slip, not on a slide. So depending on how you mount a cover slip onto the slide, if your sample's sitting on the slide, you could be 50 microns away from the cover slip, in which case your images are going to be really rather poor. So if you can grow your cells or put your tissue sections onto the cover glass, you're going to get the best image quality possible. Okay, we'll cover the confocal over the next few slides, but just to say, rather than illuminating the whole field of view at any one point, we're scanning a laser across the sample. So one of the nice things on confocal is it's not going to cause as much photo damage as you get on wide field imaging. So this is the layout of our confocal microscope, and again I've highlighted 5 key points. So again, we've got our illumination. In this case, we're using a laser rather than a bulb or a light-emitting diode. We have a structure called a pinhole, which normally sits here in front of your detector. We also have a scanner, which isn't shown on this diagram. We obviously have a detector to generate our image, but in this case, it's not a camera. It's a single-point detector, sometimes called a photomultiplier. And then finally, we're going to go through how the image is generated on a confocal, because it's slightly different. So first thing is laser illumination. Why do we use lasers? Well, firstly, they can be monochromatic, so a single wavelength, so we can get rid of the excitation filter. We don't no longer need that. The other key things are it's already a collimated beam, so a narrow parallel beam. I mentioned we're scanning that laser across our sample to build up an image. And we can get them at very high laser intensities as well, which means we can scan across quite quickly. So this is an example of an argon laser here, which is giving us an excitation output that's probably at 488 nanometers. So we've got one laser which is giving us one excitation line. And you'll see these are a lot of the kind of common lasers that people use on confocal systems, and you can see that there are a lot of molecules. These are Alexafluor dyes, excitation spectra. So you can see that they've engineered the dyes to hit the maximum for these kind of commonly used laser lines. So we covered this a little bit in the introduction to fluorescence lecture, but I'm a fan of going back over things just to reiterate. So in this case, we're using the 561 nanometer laser, and this is Alexafluor 546. On the excitation spectra, you can see we're hitting at the maximum here. If our microscope didn't have a 561 laser, said it had a 543 laser, you can see we've decreased the amount of emission that we're getting off that system just by not matching correctly the fluorophore to the laser that we have. So in red, that's our emission spectrum. This is normalized to 100%. And if we don't excite properly, then we decrease the amount of emission that we're going to get. So one of the key things when you're setting up your experiments is checking those specification sheets that we have. If you've got a transgenic line that's got EGFP, then you need to know that our system has a 488 laser which will match your fluorophores that you're using. If you have some other bizarre fluorophore, again, it's worth checking what lasers are available, how they match to the excitation spectrum of the fluorophores that you're using. So here we're about 50% emission. If we only have a 520 laser on that, then we're going even lower, down to something like 30% emission. So try and match either your fluorophores to the lasers that you have available, or match the lasers to the fluorophores that you're using. And again, that's one of the reasons we provide those specification sheets for all of our instruments. Okay, next thing is the confocal pinhole. So this is really the thing that is increasing the contrast in a confocal image. So here we've got our point detector up here. This is our confocal pinhole. And this is our excitation beam in green here, and this is our current focal plane, the red dotted line. So if we have a fluorophore in red here, which is out of our current focal plane, as that comes through the system, you'll see it's actually blocked by the pinhole. So the pinhole, you can think of it like an iris or an aperture. We can control the dimensions of that. If we have fluorescence coming from anything above or below our current focal plane, then they're blocked and they're not allowed to transmit through to get detected by the detector. So essentially we'll only see fluorescence arising from our current focal position. That's the only point at which the light crosses over through the pinhole and gets to the detector. So essentially we block out all of this fluorescence coming from above and below our focal plane. I've mentioned a couple of times now about confocal scanning. So we have our intense laser beam, and we're building up our image by scanning across the sample. So we're scanning that laser across and building an image as we go. The question is, how do we do that? So within the system, within the light path, there are two mirrors that we can control the positioning of. One controls the X position and one controls the Y position of the laser at any one time. So in this case, we've got what we call a bidirectional scan set up. So we're scanning left to right across the image, and then we're scanning right to left, back across, left to right, right to left. And as we're doing that, we're collecting photons and generating our image. Here's just an example. So we've got our laser coming off the Y scan mirror and off the X scan mirror to move the position of the laser across the sample. Next, we've got our detectors. So, as I mentioned, it's a single point detector, not a camera for a confocal system. So because we're only illuminating a single point on the sample, we only need a single point detector at the end. These have traditionally been PMTs, photomultiplier tubes. And essentially what they're doing is detecting photons and converting that into a voltage. This is a schematic of what a PMT looks like. So we have a photon coming in, it hits a photocathode, which converts the energy from the photon into an electron. We then have a series of diodes that basically amplify up that signal before we read out the signal at the end on what's called an anode. Now we can adjust the amplification of the signal here using something called electronic gain. So it's a voltage we're applying to these diodes to increase and amplify the signal. One of the downsides of this is using this increased gain, so this electronic gain also increases the noise. There's a lot of electronic noise within the detector here, and we'll cover that in a little bit as well. More recently, people have started to switch to more sensitive detectors. So these are called GASP detectors, gallium arsenide phosphide. Basically, these are about 2 times more sensitive than PMTs, and they've got a lower noise than the PMT detectors as well. They work slightly differently. This figure shows you the difference between the PMT and the GASP, so the 2 times increase is in the green-red range, but it does drop off quite dramatically at the far red range. These are really useful if you have faint samples or you're doing live cell imaging experiments. Because they're more sensitive, you have to put less light onto the samples to get an image out. And a lot of our more recently purchased systems all come with these GASP detectors. Again, that's in the specification sheets. So I mentioned about detector noise. So if you do have a faint sample or a faint signal, very often you need to increase the voltage to amplify the signal. In which case you not only get your signal, but you also get this electronic noise, this detector noise in the background as well. In confocal imaging, we get around that by performing something called averaging. So we're taking multiple acquisitions of the images and then we're averaging those over the acquisitions. So on the left here we've got an image taken with no averaging, we've got our signal, but we've also got the noise. If we take that image twice and average each pixel intensity, we can decrease the effect of that noise. And if we go to 4 times, we can really bring that back down. So we're only seeing the signal. And the reason we're doing that is the electronic noise is kind of a random effect. So it's not always going to be in each pixel at the same acquisition. Okay, so your signal should remain unless your sample's moving around. That will kind of be maintained within the averaging, whereas this random noise in the background will average down. Okay, final thing is how do we generate our image? So I've been mentioning that we're scanning our laser across our sample. Quite often on confocal systems you'll see something called a pixel dwell time. And that's essentially how long did it take the laser to scan across the sample. We're collecting photons all that time, and then we're assigning all the photons that came out of that time period to a single pixel. So essentially, although you're generating an image at the end, it's basically an array of numbers. So we're just counting photons that we've converted into an electronic signal. So you could think of it as an Excel spreadsheet of numbers, basically, that we're producing an image from. So we're scanning across, getting these intensity values, and then we're plotting them to create a picture at the end of the day. It's a bit like a mosaic. Now, one of the key things when we're generating these types of images, and this applies for both confocal and epifluorescence, is that we have pixelated images. Okay, so it's what we call a raster image. We need to make sure that we're sampling correctly to get the best image quality, or the image quality that we need out of that. So because we're forming pixels, we can potentially limit the resolution. So on the bottom here, I've got a panel. This is a single image, and I've plotted it with different numbers of pixels within each box. So hopefully you can see the most information is on the left-hand side here, where we have the most pixels. And by the time we got to the right-hand side, you can hardly see anything or resolve anything over here. There is a rule which covers this. It's called the Nyquist-Shannon sampling theorem. It basically states that your pixel size should be 3 times smaller than what you're trying to resolve. So if you're trying to look and count nuclei, then your pixel sizes need to be 3 times smaller than the size of a nuclei. Okay, so you should have a 9 by 9 block, so you can actually resolve and count nuclei. So here, for example, this is probably a little bit more than 9 by 9. We can clearly see that these are nuclei, whereas this is probably at or a little bit below 9. You know, you'd be struggling to say whether or not that was a nucleus or not. This brings up the question of what is the resolution that we're getting off our systems. Fortunately, there are equations which take us through that. If you remember me saying your resolution is a function of the NA, so that 1.4 number on the example that I gave, not the magnification. So So the equation is here for confocal lateral resolution, so that's XY dimensions. Your resolution is 0.4 times the wavelength that you're collecting divided by the numerical aperture. So at no point in that equation does it state magnification. If you look at wide field, the equation is slightly differently, so quite slightly different. So here we're at 0.61 times the wavelength divided by the NA. Put a table here which shows confocal lateral resolution. So you can choose your favorite objective. Again, we're looking at the NA here. If we go to say the 1.4, which will tend to be the highest NA we'll have on most of our systems, and we're looking in the GFP wavelengths, it's saying that our resolution is about 140 nanometers. This is a theoretical value, so it assumes a perfect system. I can assure you that there are no perfect systems. When we measure lateral resolution, it will come out more in the range of 200 to 250 nanometers. Okay, one thing you'll notice down here is we've got a 63x and a 100x, which both have 1.4 NA, which means their resolution is exactly the same. If you have this case, I would always recommend that you use the lower magnification because it will tend to be brighter. So again, brightness is a format of the numerical aperture and the magnification. If you can get away with it, an image on the 63x, you'll have to again illuminate your sample a little bit less. It collects a little bit more light on the 63x. So that's lateral resolution in XY. We also have a resolution in Z that we have to take into account as well. And what we'll tend to find is that your axial resolution is about 2 to 3 times worse than your lateral resolution. That's just the way objectives work. And now we've got another thing within the equation. So now it's dependent on the mounting media that we're using. So here we've got 1.4 times the wavelength divided by the refractive index of the mounting media that we're using multiplied by the numerical aperture. Again, so I've given examples here in air and also in glycerol. Again, we have a table in glycerol which shows us, again on the 1.4, that we're getting about 330 nanometer resolution in the Z direction. Again, that's a theoretical value. It's probably more in the 500 to 600 range in terms of actual measured resolution. So now you know you can refer to these and go, well, this is my resolution. If I'm trying to sample and get the best quality image, I need to make sure my pixels are 3 times smaller than that number. Does that make sense? Yeah. We'll cover this again on Thursday. Okay, so let's go through the experimental applications that are possible within the platform. I'm just going to bring up this table again. We're not going to go through it. Essentially what I'm going to do is highlight the different types of systems over here and give 1 or 2 examples in terms of what you can do on those different types of systems. And you'll spot that these different types are kind of grouped, so we've got multiple instruments that can do a lot of these things, and they're spread across the university, so you might not have to travel outside your building to do these experiments. So what we're going to do is look at the imaging scales from the kind of tissue level in centimeters all the way down. We're going to hit a limit at about 20 nanometers for optical microscopy. Okay, so for standard fluorescence microscopy, we can image on the scales of centimeters, so what I call macro imaging, down until we hit, until we hit this resolution limit here. So if you remember me saying our lateral resolution limit is about 200 to 250 nanometers. So essentially it's half the wavelength of light that we're using. That's basically one of the laws of physics that's not possible to get past. Unless you're very, very clever, in which case you can come up with some techniques that start to bend the rules of physics, and these techniques are called super resolution microscopy. That, as I said, can get us down to about 20 nanometer resolution. Once we're need to go beyond there, then we're going to have to use a technique like electron microscopy. I'm not going to cover any of the EM applications today. I can put you in contact with the facility manager at Bi-21 if you're interested in those. Okay, so let's start on the macro scale, and we're going to go through 4 different applications. So first we've got one called light sheet microscopy. We're going to cover multiphoton microscopy, high content imaging, and also laser microdissection. So let's start with light sheet microscopy. So here you can see an image of developing human embryo. This was done by a group in France. And these data are actually taken in 3 dimensions, so you can rotate it around and zoom in on specific features. Light sheet microscopy tends to be used in either developmental biology or neuroscience. Because it's imaging on a macro scale, you don't get very high resolution, so you're looking kind of cellular resolution quality. And as the name suggests, we're illuminating the sample with a sheet of light. So say, for example, this is our piece of tissue here. We have one objective which comes in from the side and generates a very narrow sheet of light. That's basically our excitation light. We then have another objective which comes in either from the side or from above. And basically what we're trying to do is align the focal plane of this objective, which is this red dotted line, with that light sheet. So all we're doing is illuminating a very narrow section of our tissue with the light sheet, and then imaging it immediately with the objective above. The benefit of this is we're only illuminating what we're imaging. So it means we don't get any extra bleaching like we do on the confocal and the epifluorescence techniques. Some of the downsides is a lot of the time we need to immerse this imaging objective into some kind of media that's holding the sample, using what we call a dipping lens. And also the sample needs to be quite clear. So if we're imaging tissue on a centimeter scale, then we need to be able to get the light through that to be able to visualize it. So the reason it's used a lot in developmental biology is people use it on things like Drosophila, zebrafish, embryos, things like worms that are inherently clear already. You can use it on larger tissue samples, but we need to chemically clear those tissues out to make them clearer for the imaging. You guys might have heard of some of these techniques. Something like clarity, which we've got an example of up here. So this is before clarity clearing, this is a mouse brain, and then it's still there, but it's been chemically cleared out. So tissue clearing has been around for a very, very long time. So it was first developed in 1914, and there's 2 main different methods. So using either organic solvents, in which case we're removing water out of the sample, and then we match the refractive index of the tissue with some kind of organic solvent, and that allows it to become clear. One of the downsides is this isn't always applicable with fluorescence proteins. And some of these techniques you might have heard of are BAB, which is fairly nasty stuff. Some of the disco techniques. One that quite a lot of our users are using is ethyl cinemates. Smells of cinnamon, which makes it quite nice in the lab. Apparently you can drink it, but I've never seen anybody do that. I'm not sure why you would want to do it. The second method is aqueous-based methods, and these work slightly differently depending on the method that you're using. So something like clarity works by removing lipids out of the tissue, and then followed by refractive index matching. Some of the other methods, something like CDB, is basically an immersion method. You basically just put in something with the refractive index that matches the tissue. It soaks in and needs to go clear. It tends to take a bit of time. And the downsides on the tissue clearing is It can be tricky. It's not the type of thing you're going to decide you want to do tissue clearing and then next week you're going to have your samples perfectly clear. It takes a bit of time to develop the techniques for your specific tissue. And some of the techniques can be very, very expensive. We have our light sheet systems in the lab in this building. This is one of the images which was taken on that system. This is a cochlear. And here the researchers were looking at a 3-color imaging experiment. They're now pushing up into 4 colors. And they're actually looking at cochlear implants and how they affect the tissues around them, how much scarring goes on when you do a cochlear implant. So it can be very, very beautiful data sets come off the light sheet. One thing I should say is because we're only illuminating what we're imaging, we can scan quite quickly down. This is a piece of tissue which is probably just under 1 centimeter deep. That was probably imaged in about 12 minutes. If you tried to do that on a confocal, you would set it up and then you might have to come back 4 days later. It would take so much longer to acquire the data. One of the downsides is because we're taking thousands and thousands of slices through this, is the data sets can be quite large as well. So this is probably a 30 gigabyte file just for that single acquisition. Multiphoton microscopy. Okay, so this is used for what we call intravital imaging in live animals. In this case, we use infrared wavelengths in the kind of 700 to 1,000 nanometer range to excite our samples. So if you remember from the first lecture, this is our Jablonski diagram looking at the energy levels of the electrons in our fluorophore. They're normally sitting at the ground state, and when we put in our excitation light, the fluorophore can absorb the photon and jump up into the high energy state. Yeah? In multiphoton microscopy, what we're doing is we're putting in pulses of laser light, excitation light, and we're relying on 2 photons being absorbed by that molecule at the same time. Now, they have a longer wavelength, which means they have less energy, but if they hit the photon at the same time, those energies basically add up, and we can still get the same effect. So the fluorophore becomes excited, and then decays back, and then emits light back out in the kind of green wavelengths. Why do we do that? Why is the longer wavelength important? Well, it's important because light of longer wavelengths can penetrate through tissue deeper into the tissue, so it gets less absorption, less scattering. It also causes less damage to the tissue as well because there's less energy in those photons. So if you've got a GFP molecule, normally with your confocal excitation, you're exciting, say, a 4-8 laser or something around the 490s. Into photon excitation, you're pretty much doubling that up, so you're exciting it with something like 920 nanometers. What can we use that for? So here we've got an example from a group in microbiology who are looking at herpes infections in mice skins. They're imaging multiple channels here. So in blue they've got collagen, in purple they've got the herpes infections, and then in green and red they've got 2 different types of killer T cells. So green, the ones which are specific to herpes, and red, the ones which aren't specific to herpes. And what they're able to do is image these cells moving around in the skin over long periods of time. And using a software called Amaris, they can track these and look at, what's the average displacement, what's the average speeds, and get a lot of quantitative readouts in terms of how these 2 different cell types are behaving within the skin. So anyone that wants to use the multiphoton microscopy, it's actually done via collaboration with Scott Miller's lab in the Department of Microbiology and Immunology. This is showing skin, but they also do a whole heap of other tissues, regularly imaging in brain and lymph. I think they're also moving into retina as well now. Essentially, if you can find a way of doing the surgery to get the sample underneath the microscope, then you should be able to image on the multiphoton system. Now I said we can penetrate deeper into samples. We have a limit probably of 500 microns. You can push to 1,000 depending on the tissue, but you're not going centimeters into the sample. So. Next method we're going to cover is something called high content screening. Again, our high content screening system within the platform is here in the medical building. This is the Operator system, and basically it allows you to do wide field and confocal imaging of either fluorescence, bright field, or phase contrast, and you can essentially image cells or tissues or organisms as long as they'll fit in a microtiter plate. So a 6-well plate, anything up to a 1,536 well plate, we can image automatically on the system. So we can basically say, image these channels, image these wells, do it every 20 minutes, and tell the system to go off and do that automatically for us. So again, it's not massive resolution, so we're not looking really at high resolution information. We can get subcellular information, but it's a little bit limited. But it allows that kind of automation in the experiments that you're setting up. The system is capable of live cell and time-lapse imaging, so we have it attached to CO2, and we can temperature control as well. So we have people running experiments for 5 days quite happily on this system, waiting for a rare event to occur. And one of the real powers of this system isn't just the automation, but it's also the downstream analysis as well. So we can also tell the system, you know, go and count nuclei and plot it out to me in an Excel spreadsheet. We can also tell it to do a lot more complicated things as well. There are machine learning algorithms within the software, so if you don't know the difference between the two cell types, but you can visually say this cell's, you know, in one state, this cell's in another state, we can teach the software the difference between those two different cell types just by clicking and saying, that one belongs to that group, that one belongs to that group. So some of the applications for that are things like RNAi screening, drug screening, stem cell research, and whole organism imaging. I don't mean mice, I mean things like worms, zebrafish, fly, embryos, things like that. It's really hard to squeeze a mouse into a microtiter plate. This is one example from a group in clinical pathology. So they have these cells which are called Lego cells, so they're basically cancer cell derived, and each individual type of cancer cell has its own individual color. So they're doing long-term live cell imaging on these samples and looking at how these different cell types interact. So do they always only want to be next to the cell of their own type, or are they fairly gregarious? How are they sharing nutrients? So they're looking at kind of tumor microenvironments, but in a 2D setup. So here they were able to segment out and look at how each of these cells behave in a mixed population. Last technique in this section, in the macro section, is laser microdissection. This system again sits here in the medical building. It isn't massively used, but it's basically a technique where you can isolate cells or regions of cells and then perform downstream omics analysis of things like genomics, qPCR. So in this case, if you can use Paint, the Microsoft program, you can use this microscope. So you're just basically drawing around the region that you want to excise out. And then we laser etch that region. And then we have these little blue dots. That's a laser catapult. So we're firing a laser and basically removing that piece of tissue from the slide. There's a little cap from an Eppendorf tube that sits directly above the slide, and it's catapulted back up and caught into the liquid sitting in the cap. So here you can see we've etched around this region and we've excised out all of this tissue, which we can then go on and do different analysis on. So normally they have to be cryopreserved samples for this to work, especially if you're doing any RNA-based work, so they need to be fairly fresh samples. Next technique is wide field. So as I mentioned, we don't tend to have very many wide field systems. There's currently one, which is based at Bi-21. And in this case, the reason we have it is it has a deconvolution module attached to it. I'll cover on the next slide what deconvolution is. But this is really ideal for very thin, cleared samples, such as cultured mammalian cells. And again, they need to be sitting on the cover slip. The system we have at Bi-21 can be used for live cell imaging, although I probably wouldn't recommend it. If you remember wide field imaging, you're illuminating everything, so you're going to be causing a whole heap of phototoxicity to your samples. If you're trying to do long-term live cell imaging, it doesn't tend to work very well. Here we've got an example of a standard wide field image. This is a malaria parasite, and then this is the same data set which has been deconvolved. So you can see it's increasing the image quality over the raw data. So what is image deconvolution? So essentially it's a mathematical restoration method which is going to increase your resolution in all 3 dimensions, in X, Y, and Z. Increase your contrast and remove noise. And the question is, how is it going to do that? So essentially what we do is we can take an image of a single point, say a fluorescent bead or something on the microscope, and we can look at how that behaves when we image it through 3 dimensions. It's what we call the point spread function. If we can measure that, we can then model how light behaves in the microscope. And what the algorithm does is goes back, looks at fluorescence and says, at this focal plane, this object is in focus, so if I go above and beyond that, there should be a little bit of light which is spread out from that point. So it's mathematically reassigning light that may be out of focus back to where it originated from, and that's how it's increasing the contrast. So it takes those fuzzy images that you tend to get on epifluorescence microscopes and turns them into much, much sharper, more like confocal quality imaging. What do you require for image deconvolution to work? So first of all, you need that Nyquist sampling, that 3 times smaller than the resolution sampling. And you also need a Z stack of data, so it works best if you've got information above and below the focal plane that you're looking at the moment. How can you guys do that? We have 2 licenses for something called Huygens, one here in the medical building and one based down at Bi-21. And the nice thing with this software is it has deconvolution wizards which take you through each of the steps. We can do that for wide field, for confocal, for area scan, which we'll cover in a few slides' time, and also for light sheet data. Because the data sets can be fairly large, we have GPU acceleration, so it does all of the compute on the GPU rather than the CPU. You can also set up a batch processor, so you can say, go to this folder and perform a deconvolution on these 100 images that I've collected. We'll do things like time-lapse correction, so if you're bleaching, samples bleaching, it's drifting in Z, changes your background, cleans up your imaging. This PSF distiller is that technique where we're acquiring that single point image to act as a model for the deconvolution process, and can also correct for a whole heap of other aberrations as well. So if you want trained on how to use the Huygens Professional, let us know and we can show you how to use the software. TIRF microscopy. So TIRF is total internal reflection microscopy. In most other fluorescence modes so this is an inverted system, this is our cover slip, this is our sample. Our objective is below here. We are taking the excitation light and shining it directly up through our sample. We're collecting, exciting the fluorescence, the fluorophores, and then collecting the fluorescence that's been emitted straight back down. In total internal reflection microscopy, we illuminate through the objective at a critical angle, which means that the majority of the light is reflected across. this produces is what we call an evanescence wave. So just the first 100 nanometers past the cover slip will actually get excitation light. So just the molecules right at the very interface with the cover slip will become excited. This has real nice benefit. It gives you a very high signal to noise in that single region just past the cover slip. But it does mean you can't image things like nuclei or anything on the top of the cell. Here's an example of some turf imaging. We now have 3 systems in the platform that are capable of turf imaging. One of those we have 4 cameras on, so we can actually take 4 turf images simultaneously. Some of the applications for turf imaging are things like calcium flux imaging, cytoskeletal imaging. If you're interested in mitochondria, they often sit within that gap, that 100 nanometer gap at the bottom of the cell as well. Okay, so that was kind of macro scale imaging. The next thing we're going to cover is in the kind of cellular, subcellular region. So we're going to cover from 10 microns down to the 20 nanometer, 20 nanometer range. So we're going to cover confocal microscopy and things called F experiments, and then we're going to go through some of the super resolution techniques that are available to you guys as well. Okay, so confocal, there's a lot of content here that we could go through, so I'm just going to show you a few kind of key highlights. First of all, a lot of our confocal systems come with automated stages. That allows us to take data in 3 dimensions, so not just X, Y, but also in Zs as well to build up 3-dimensional images. It also allows us to use the XY stage to do things like multi-point visiting experiments or tile scans. I'll come on to tile scans in the next slide. We do a lot of dynamic imaging on confocal systems as well, so things like live cell imaging, the F experiments, which we'll come on to, which are looking at protein dynamics or protein-protein interactions. We can also do cool experiments like photoconversion. So if you remember from the first lecture, I told you about photoconvertible fluorescent proteins, where we can switch them from one emission to another emission. And we also have some confocals which are equipped with specialized detectors, some for super resolution microscopy, and some for spectral detection of fluorescence as well. So over the next few slides, I've got a few different examples of some of those points that we've just gone through. So first one is 3D data acquisition with tile scanning. This is what looks like a cross-section through a spleen at a fairly low resolution. If I was to zoom into that image, you would see much, much higher detail. So you could image that fairly quickly on, say, a 5x objective, but you'd be limited in terms of resolution. What this lab here have done has taken a higher magnification lens, so they've actually only acquired a field of view something like this. And they've used the automated stage on the system to actually take an image, move across, take an image, move across, take an image, move across. And so they've moved the stage across in XY to build up a higher resolution, higher quality image of this whole region. And you can also do that in Z as well. So you can build up a large 3D model of the tissue that you're interested in. I've seen some labs presenting data sets which are 1 terabyte in size, massive high resolution information over whole organs. So you're kind of limited by your imagination in terms of how much you can do. Next thing is spectral unmixing. So we have some systems which have 32 detectors aligned in an array, and what we can do is basically spread the visible light spectrum across those 32 detectors. What that allows you to do is put a lot more fluorophores onto your samples, and using a technique called spectral unmixing, we can basically define each pixel to a certain fluorophore. So in this example here, Darryl Johnson has 9 different fluorophores on this piece of tissue, And he's used spectral and mixing to go back and assign each individual cell to say what different protein makeup that cell has. There are also commercially available kits, so things like Optical, I'm not sure you guys may have heard of, which also allow you to do these multiplexing experiments as well. So yeah, Darrell's got to 9. I know of one group that's got to 10 different fluorophores, and we can spectrally separate those out quite nicely using those spectral detectors. In terms of live cell imaging, the best system we have currently in the platform is a spinning disk confocal. So that works slightly differently to a laser scanning confocal. So if you remember, the confocal I've been talking about before is scanning a laser across your sample to build up an image. On the spinning disk microscope, as the name suggests, we have a disk that spins, and it has lots of different pinholes. In this case, the acquisition is going to be much, much faster, and we're also detecting on a camera, not on a PMT like you are for a standard confocal. One of the downsides with the spinning disk system is it's not really applicable for thicker samples. So again, for cultured cells, it's probably a bit better. Let me just show you how the spinning disk works. So we're illuminating here, light coming through the spinning disk onto our sample, and when that's spinning really quickly you can see it's even illumination, but if I slow down the rotation speed you can see the light coming through each of those individual pinholes, but this disk spins really, really fast, so you don't actually see that illumination. So again, it means you're not illuminating the full sample all the time. There is a chance for some of the molecules to come back to the ground state and kind of rest. This is one example of some of the data. These are cells undergoing multiple rounds of mitosis. This is can be very technically difficult because cells will tend to hit a cell checkpoint. If there's too much phototoxicity going on during these experiments, they'll just hit a checkpoint and stop dividing. So you have to be very, very gentle on these cells if you want to do live cell mitotic assays. Okay, next thing we're going to go through are what we call the F experiments. These broadly look at protein diffusion and protein interactions, and there's a number of different F experiments. You'll find microscopists love an acronym, so everything has its own different acronym. So we're going to go through fluorescence recovery after photobleaching, or FRAP, fluorescence loss in photobleaching, which is FLIP, fluorescence fluctuation spectroscopy, which is FFS, and fluorescence resonance energy transfer, which is FRET. Sometimes this is named Forster resonance energy transfer after the guy who invented the technique. So we'll go in order. So we'll start at FRAP. So what are we doing in FRAP is we're going to take our cell and we're going to selectively bleach a region within that cell, and then we're going to look over time and see how the fluorescence recovers back into that space. So we're laser scanning across. When we hit the region that we're interested in, we tell the system to go up to 100% laser intensity and photo bleach all the fluorophores which are in that space. Here, we don't have any fluorescence left remaining there. And then over time, we'll just go back to an imaging, and we're looking at new molecules that are transiting into this space. Okay, and if we look at the fluorescence intensity within this region of interest and plot that, we can see pre-bleach, which is up here, we've got a flat line. When we bleach, take the intensity right down, like in example B, and then over time we'll get a recovery as new molecules transit into the space. So what we're looking at basically is the mobility of those molecules, the ability of them to transit into the region that we've photo-bleached. That can give us a number of quantitative readouts. So we can read out, for example, the half time to recovery. So the t-half gives you an idea of the mobility of the molecules. We can also read out a ratio of immobile fraction versus mobile fraction. So what's the percentage of molecules which are actually mobile? when would you use this technique? So there's a few examples here, looking at things like membrane protein dynamics. So again, bleaching out a labeled syntaxin 1 here, and then over time looking and seeing how that molecule moves in from other parts of the membrane. It's commonly used also looking at nuclear translocation, so you can go in and bleach the nucleus and then look how long it takes for molecules to transit across the nuclear pore complex into the nucleus. Fluorescence loss in photobleaching is a kind of similar technique, except rather than doing one bleaching step, we're doing multiple bleaching steps. So here we're doing multiple bleaching steps within our region, and what we're looking at is how interconnected the regions are. So here we can see the cytoplasm's bleaching out much faster than the membrane here, which makes you think that the cytoplasm's in more direct contact with the region of interest in the membrane. So you can use it for looking at things like continuity of membranes, continuity of organelles. Fluorescence fluctuation spectroscopy, so FFS. These are, again, a group of methods which investigates the mobility of molecules. These will tend to be done on a confocal microscope. Within our platform, we tend to use the Olympus FV3000 for most of these. And these can provide readouts on molecule concentration, the diffusion coefficient, the number of molecules, the oligomeric state of the molecules. So depending on how you approach these sets of experiments, you can get different readouts. For example, so fluorescence correlation spectroscopy, here we're parking our excitation beam from our fluorescence in one position, and we're looking at molecules coming in and out through that volume. This gives us this kind of peaks and troughs as molecules come in and out of that volume that we're exciting. If we do a correlation curve, we can look at what the diffusion and get a readout of the size of the molecules based on this pattern. We can also do raster scanning, so confocal scanning, and do something like a numbers and brightness analysis where we can look at what's the oligomeric states of the fluorophores that we're looking at. Again, these are really large, different number of techniques, and we don't have time to really go into them today, but if you're interested in any of these things, then one of those techniques might be applicable to you. Okay, the last F experiment we're going to cover is fluorescence resonance energy transfer, or FRET. In this case, I think I covered this a little bit in the introduction to fluorescence microscopy. We've got two fluorescent proteins here attached to different proteins. This one, in this example, is a sensory domain. This is our substrate. When these things are separated, it means that these two molecules are greater than 10 nanometers apart. If we excite the CFP molecule here, then we'll get emission in the blue wavelength, so here at 475 nanometers. If this complex forms, so these two substrates and sensory domain interact, this brings these two fluorescent molecules within that 10 nanometer range, which means when we excite the CFP molecule, rather than emitting in the blue range, it's going to transfer that energy to the YFP molecule, and we see the emission in the yellow wavelength, so 528 nanometers. Okay, so we're getting a readout of the proximity, the interactions between these molecules through this FRET, and how the emission changes. In order for FRETs to occur, what you need is a donor, which in this case is the CFP molecule, and an acceptor. And what you really want is as large a spectral overlap between the emission spectra of the donor and the excitation spectrum of the acceptor. Okay, that's going to give you the most efficient energy transfer between those two molecules. What are the applications, possible applications for these? So I mentioned protein-protein interactions. Excuse me. here we've got our two molecules which are separated. When they bind, we'll get frets occurring, but not before. We can look at protein-substrate interaction. So here we've got a calcium binding site. When there's no calcium bind, the two molecules sit apart. When calcium binds, it causes structural rearrangement, and these two are close enough for frets to occur. We can look at kinase activity. So again, a phosphorylation recognition domain, which will cause a structural rearrangement, allowing frets to occur. And then some of you guys may have been doing fret assays and didn't even realize. So this is a protease assay kit with a protease recognition sequence. Here the two molecules are close enough for fret to occur. If you get cleavage, then obviously they get separated and you get less fret occurring. Now when it comes to analyzing fret, there are a lot of different ways of doing this. These are some of the most commonly used ones. So one is called acceptor photobleaching. We would recommend this for fixed samples only. In this case, we've got our two molecules, and here we can bleach out the acceptor, which causes an increase in the intensity of the donor, because it's not transferring its energy if there's no molecule there to absorb. So we can do a bit of mathematics here and work out what the fret efficiency of those two are. We can do simple ratiometric measurements between the two fluorophores, but that can only be with fret pairs with a fixed stoichiometry. So if we go back to some of these previous techniques where you've got two molecules which are on the two fluorescence molecules which are on the same molecule, you know that's a one-to-one stoichiometry. Sensitized emission is probably one of the more clean ways to do it, but it needs a lot of control and a lot of mathematics. I'm a biologist by training. Mathematics scares me. I don't like sensitized emission. The cleanest way of doing any fret analysis is to use fluorescence lifetime. In this case, it's completely independent of wavelengths, and we'll cover fluorescence lifetime in a couple of slides' time. Okay. So fluorescence lifetime imaging microscopy, or FLIM. As I mentioned, you can use for what we call FLIM frets analysis. One of the key things with fluorescence lifetime imaging microscopy is normally when we're doing fluorescence microscopy, we're collecting emission over a specific set of wavelengths. So we've set a bandpass filter or a range that we're collecting fluorescence over. With fluorescence lifetime, we're not worrying about what the wavelength of the light is. We're looking at the lifetime. And if you remember from the introduction to fluorescence, the lifetime is how long your fluorophore stays in that excited state before it returns back to the ground state. So one of the nice things is proteins or fluorophores, which are very, very similar in terms of emission wavelengths, can have very different fluorescence lifetime. So here we've got an example of alexafluor-488 and EGFP, which sit spectrally right on top of each other, have very different lifetimes. So alexafluor-488 is 4.1 nanoseconds, and EGFP is 2.6 nanoseconds. So that allows us to separate those. Some of the other applications are things like multispectral analysis. So if you remember, we go back to those experiments where we've got 9 different fluorophores. We can actually separate those out as well using fluorescence lifetime. We don't have to spectrally unmix. People also use fluorescence lifetime for looking at autofluorescence as well. So different bits of tissue, as this example here, plant tissue, have different fluorescence lifetime species. There are 2 ways that we can do fluorescence lifetime. So traditionally it's been done in what we call time domain. So we've got our excitation pulse, and then we look at the fluorescence that's coming off. In this case, we need to fit this curve to work out what the fluorescence lifetime is. The system that we have works in a frequency domain, which works very, very differently to having to fit these curves. And we plot our fluorescence lifetimes on what's called a phasor plot. So this is essentially in Fourier space. We have a point here, so the fluorescence lifetime should always pretty much be on this circle. If they go around to the clockwise, then they go shorter in lifetime. If they go around anti-clockwise, then they're increasing in lifetime. One example of this is a group at Bi21 who have a fret biosensor looking at histone compaction. So they've got histone 2b labeled with EGFP and histone 2b labeled with M-cherry. And when the histones aren't very compacted, you get no frets. And as they become more and more compacted, your fret signal increases. And we can actually separate out these three different species on this universal circle. Okay, so it's a very neat, cool way of looking at different protein-protein interactions or any of the other assays that we covered for frets. So that's looking at dynamics of individual molecules. Let's now switch to some of the super resolution techniques that are available to us. I've given a quick overview table here just to remind you where we sit. So on standard confocal, we can image up to 50 microns in depth. Our standard lateral resolution is about 250 nanometers. In Z, it's about 600 nanometers. These are some of the commercially available super resolution techniques. So ARIScan I will cover in a few slides' time. STED, which is stimulated emission depletion, we don't have within the platform, so I'm not going to cover that, but that can get you down to 40 nanometer resolution, or if you want isotropic resolution, 130 in X, Y, and Z. 3D Sim we have, which gives you about a 2-fold increase in resolution. And then finally, single molecule localization microscopy as well will get you down to about 20 nanometers resolution. Okay, so let's go through the confocal super resolution techniques. So within our platform, we have now 3 systems equipped with the ARIScan detector. And what this does is basically makes use of the pinhole. So people have known for a long time that if you narrow the pinhole, you increase your resolution. So this figure here shows as you narrow down your pinhole, you can increase your resolution. The standard setting for a pinhole on a confocal is what we call 1 area unit, so this position here. The problem is if you go down to 0.2 of an area unit, you're blocking out 95% of your light. So it's very difficult to actually get a good quality image when you're blocking that much light. What the ARIScan detector does is it doesn't narrow down the pinhole, in fact it actually widens the pinhole out a bit. But rather than having a single point detector, it now has this 32 detector array set up as like a compound eye, like a fly's eye. And the reason they're doing this is each one of these individual detectors now has an equivalent diameter of 0.2 of an area unit. Okay? So you're getting the effect of narrowing the pinhole without actually narrowing the pinhole. There's a bit of clever mathematics goes on to extract the information out. So this is an example of an image taken on the same system in confocal modes versus the ARIScan mode. So the ARIScan, I should have said, gives you about 2-fold increase in resolution, so you're looking at about 120 to 140 nanometers resolution. And you really don't have to jump through many hoops in terms of sample preparation or anything to get this increase in image quality. Now, it doesn't look massively startling until you start to look at some of these femto patches where you can't really see what's going on here, whereas if you compare it in a confocal image, you can start to really visualize what's going on in smaller detail. So the Zeiss systems that have the ARIScan detector have their own processing algorithm. We also have, as part of the Huygens deconvolution package, an ARIScan module which can increase the resolution we can get off the Zeiss system. So this is one example of some imaging we've been doing recently. In the top panel here, this is ARIScan processed data. So we're trying to look at these microvilli on this cell here. If we use the Huygens ARIScan deconvolution, we can increase the resolution down to 90 nanometers from 120 nanometers. structures. So you can actually see we start to resolve these structures a little bit better. We've got much nicer contrast going on in these channels here as well. So it doesn't seem like a massive difference, but it makes quite a startling difference when it comes to your image analysis. Next technique we're going to use is 3D structured illumination microscopy. This is essentially a wide field fluorescence technique. This will produce an 8-fold increase in resolution, X, Y, and Z. We can do up to 4 color imaging, but the downside is we need to take 30 more exposures than on a confocal microscope. And the reason we need to take 30 more exposures is we image our sample through an interference pattern. And we move that interference pattern across our sample, and we can extract higher resolution information from that interference pattern. These are some examples of data taken with our 3D sim system, which is based at Pi21. On the left-hand side here, we've got a wide field fluorescence image. These are Hendra viruses, which is apt today as they infect horses. This is our 3D sim image, and essentially we're looking at a viral coat protein here. We can see the diameter of these viruses. I think from memory they were in the order of about 280 nanometers across. We measured them in fluorescence, and that agreed pretty well with the data that the researchers had from electron microscopy. This is a malaria parasite, again in wide field mode versus 3D sim mode. And then we have microtubules in a eukaryotic cell as well. Here we go, this is another example. So this is 4-color super resolution imaging. So this is another parasite called Toxoplasma, and this is the invasion complex, the part that attaches to the cell and forces the parasite into the cell it's going to infect. This group had identified a protein and had tagged the N and the C terminus of this protein. You can see we can actually resolve the N versus the C terminus of this protein, this ring 2 protein. It just happens it's a megadalton protein that lines up in these long strips as a large kind of barrel. One of the nice things we were able to do was to show that when the cells were intracellular, so before they'd extruded out, the proteins were in one orientation, and when they became extracellular, they actually flipped out. So there was some kind of mechanistic change in terms of the orientation of this barrel structure. We were feeling pretty happy with ourselves after generating these 3-color images and sent it off, and the reviewers said, well, that's nice, but we don't know what these are doing in context to some of the known markers, so they asked us to do a 4th color, which we have here, so 1, 2, 3, and then the 4th color is the full parasite. So 4 color is pretty technically difficult to do in 3D sim. The last super resolution technique we'll cover is single molecule localization microscopy. So one of the issues we have with fluorescence microscopy is you're normally illuminating your sample and you're seeing all of your fluorophores emitting back at the same time. Okay? So it's very difficult to resolve individual molecules. The guys who developed these techniques won the Nobel Prize for chemistry a few years back. There are 3 main methods, although there are other acronyms available as well. So PALM is photoactivated localization microscopy, STORM, stochastic optical reconstruction microscopy, and PAINT is point accumulation for imaging in nanoscale topography. In these single molecule experiments, what we're trying to do is not illuminate all of our molecules at any one time, but only illuminate a few molecules. This movie of the Eiffel Tower in Paris shows you what we do for this. So here we're trying to induce just a few of the molecules to emit at any one time. That means we can be sure it's coming from a single event. It means we can actually fit that to a Gaussian distribution and say this is the center point of that molecule. So these are our original images here. This is what we've detected, and then over thousands and thousands of frames we can reconstruct a high resolution image. So these different techniques have different ways of causing this blinking effect. So in PALM we're using photoactivatable fluorescent proteins So proteins which will either go from an off to an on state or switch from, say, a green to a red state, and we can control that switching frequency. In STORM, we're using some clever chemistry and some high-powered lasers to basically push most of the fluorophores into a long-lived dark state, where they're not going through that cycle of excitation and emission. They're sitting in a triplet state where they're not doing anything. So only a few of the fluorophores will come back, get excited, and emit. And in DNA Paint, we're looking at the binding, the stochastic binding of DNA oligos. So you've got an antibody that's detecting your protein, an oligo, which has our fluorophore, and there's a little oligo tag sitting off your antibody, and we're looking at the binding on and off of your oligo onto your antibody, essentially. So it's producing that blinking effect. Here's an example of DStorm experiments. So again, this is malaria cells imaged in wide field mode on the left, and then using DStorm, these images were taken from 15,000 individual images, reconstructed back to give us this higher resolution information. Okay, so one thing I haven't really covered but we'll cover tomorrow that we also have on the platform is image analysis. So we have licensed softwares like Amaris and Huygens that I've mentioned a bit, and Velocity. Also Metamorph, which I don't use very often. We do quite a lot of work with Fiji or ImageJ. As I mentioned, we've got our Fiji workshops this afternoon and on Thursday as well. I'll cover this image analysis a little bit more in Thursday's lecture. Just to go back over what we've covered in this section. So if you're looking at macro imaging, then we can use the light sheet, the multiphoton, the high content imaging, or laser micro dissection. If you're looking at subcellular, then we can go to super resolution or some of the confocal techniques, so ARIScan, 3D SIM, single molecule, fluorescence lifetime imaging, or things like fluorescence fluctuation, spectroscopy. You remember all those F experiments. If you want to go beyond that, then you need to go to scanning or transmission electron microscopy, tomography, microanalysis, single particle cryo-EM is the buzz technique at the moment in EM. Okay, so with that we're done for today's lecture. So we've covered what the platform is, we've covered the anatomy of a wide field or epifluorescence and a confocal microscope, so hopefully now you've got a better understanding of those two different systems. And then you've learned about some of the cool stuff that we can do within the platform. Hopefully some of it applies to your research. If you want to know more about a specific technique, then please let us know. I just wanted to quickly highlight a few upcoming events between now and the end of the year that we have. So we have our Fiji for Beginners workshop today from 1 o'clock, if I have any voice left. On Thursday, again from 1 o'clock, we have the Fiji for Quantification. So Fiji for Beginners is how to open your data, how to merge, split images, how to present 3D data, time-lapse data in presentations and things like that. Fiji for Quantification is really, you've got an image, you want to quantify it, you want to count the nuclei, you want to look at the intensity, you want to segment out individual cells and say what the intensity is in each individual cell. So really more advanced stuff. On Friday we have an advanced fluorescence imaging symposium. We've got the guys coming from 3i to tell us about some of their cool toys for imaging high speeds, live samples or whole tissues, whole organs. Next week we've got the Fiji macro writing for biologists workshop. So if you're already using Fiji, you'll know that you have to click lots and lots of buttons. We can write macro codes to do things automatically for you. So Ellie from the team runs this two half-day workshop which takes you through how to do that. If you're interested in that, you can sign up on our webpage. And then on the 3rd of December, we have Leica coming in to run a demonstration on their new Thunder system. It's essentially a wide field deconvolution system that works really nicely on thick samples, so up to 500 microns into samples. You can get really high quality imaging. Okay, with that, thank you. Hopefully see you guys on Thursday. If anyone wants to have a quick look through the lab, then let me know. I'm happy to take you guys through. Thank you.